Hi all, welcome back. We're going to start discussing blood. Okay, and so blood is the kind of a liquid version of a connective tissue. Okay, and components of blood are things like red blood cells, white blood cells, platelets, plasma, etc. So we're going to dive into it. We'll break it up into different parts and hit blood as this very important connective tissue. So first off, blood itself is the only connective tissue that has a liquid matrix, and that liquid matrix we call plasma. Okay? And so when we're talking about blood, it's super important for homeostasis. It can maintain ion balances, it can maintain nutrients, it can uh, transport hormones and maintain hormone balance, changes temperature in the body. So lots of things that are going on, and really, it's from a connective tissue perspective, it's super important for homeostasis. Okay? And we'll look into why that is. Okay, about 8% of an individual's body mass is blood. And so females being smaller than males, normally we're talking 4 to 5 liters. Males, we're talking 5 to 6. So some of you might be thinking, well, for a blood transfusion, maybe the individual needed 1 or 2 units of blood. That unit is referring to pints, okay? Because we measure blood in this case in liters, okay? A pint, there's about 2.1 pints per liter. And so if someone's down 2 units of blood, they get roughly 2 pints of blood. Or they were, sorry, if someone's down 2 units of blood, okay, then they get roughly 2 pints of blood or 1 liter. Okay, so that's just puts that kind of in perspective when you hear about blood transfusions, or if you're giving blood at a blood bank or whatnot, and they would say, well, we're going to take, excuse me, a single unit from you. Okay, they're going to take a pint. Okay, and so if someone was down 2 units of blood and they're a female, they might be down 25% of their blood. Okay? And so that can be a huge amount of and be some major problems for that individual. And we'll kind of talk about how that works later. All right, so blood cells themselves, whether it's white blood cells, red blood cells, the initial phase of the blood cell is formed in the red bone marrow. Okay? And so that's what we call the formed element. And we'll come back and talk about how this works. The formed elements are one of these 3 things. So red blood cells, normally abbreviated RBCs, white blood cells, WBCs, and platelets, which you might see them written as thrombocytes. Okay, but these are cell fragments and involved in clotting. And we'll come back and we'll talk more about clotting in later lectures. All right, so when we look at blood in general, a single drop of blood, about 55% of it is that liquid base, okay, which we call plasma, and we'll get into that, okay? The other 45% is formed elements, with that being dominated by red blood cells, at least from a human perspective, and well, a mammal perspective, okay? Platelets, about almost 5%, okay? So that leaves pretty little for white blood cells. White blood cells, though there's quite a few different types of white blood cells, neutrophils, eosinophils, basophils, monocytes, lymphocytes. Neutrophils are by far the most common in humans. Lymphocytes next, and then eosinophils, basophils, monocytes. Roughly 1-3%. It depends on the individual, but not very much. Okay? Jumping back to the plasma, okay, that 55% of blood, most of it's water. Okay, you have the electrolytes, sodium, potassium, calcium, things like that. You have some proteins, albumins, globins, fibrinins, We'll come back, talk about the purpose of these. You got some waste products normally associated with the breaking down of biomolecules like lipids, proteins, nucleic acids, carbohydrates. Some of those waste products that are more solid would fill here. Some of the waste products that are more gases would fit in the gas category, things like nitrogen, carbon dioxide, and then of course oxygen being needed for cellular respiration. And then the nutrients, vitamins, and we just finished talking about hormones, and the hormones that are involved in plasma are very important. Okay, so what about a centrifuge blood sample? So many of you guys have probably been through this process. You go get your blood drawn, they centrifuge it, and what does it tell them? Or what does it tell someone who centrifuged it? Well, lots of things actually. So the first part of that centrifuge tube, which will be the top layer, is typically kind of clear to yellow color. That's plasma. Okay? And based on the plasma level or the ratio of plasma level maybe to red blood cells, that can tell you can tell, well, is the individual anemic? Do they have enough red blood cells to support their body? Okay. Red blood cells is the bottom layer, which we call hematocrit. Okay, so that's the percent of red blood cells, and it's packed red blood cells. And so a centrifuge is just spinning really, really fast, and it separates things based on mass. And so things that are heavier are pushed to kind of the bottom. And so red blood cells having hemoglobin inside them, that molecule is fairly heavy with bound to iron. It pushes it down to the bottom of the centrifuge tube, and then the layer between these two, between plasma and red blood cells, is your white blood cells and your platelets. Not very much. It's kind of a buffy coat. And in later lectures, I'll show you an actual centrifuge tube, but here's kind of cartoon. Plasma is that top layer. The buffy coat, that small layer in between, sometimes it's gray. Okay, that's your white blood cells, platelets, and then your hematocrit's that bottom, that 45%. So by measuring how much plasma there is, ratio of plasma to red blood cells, or even white blood cells, sometimes if the white blood cell layer is thick, it'll indicate that the individual has an abundance of white blood cells. Maybe they're sick, maybe they have a parasite, maybe they have cancer, like leukemia or something like that, which is going to produce lots and lots of white blood cells. Remember, cancer is just uncontrolled cell growth. And so if you have cancer of white blood cells, then it's just kicking out lots and lots of white blood cells. And so you might have a really thick layer, excuse me, of white blood cells there. Pardon me. Okay, so the formed elements, okay, how are they formed? Well, that process is called hematopoiesis. Hematopoiesis is the generation of hematopoietic stem cells. These hematopoietic stem cells are also called hemocytoblasts. Normally, in my opinion, you hear hemocytoblasts more often. But either one, either term's fine. And really, that's the elements that are going to build kind of the non-liquid portion of blood. Okay, so your red blood cells, the white blood cells, and the platelets. That hematopoietic stem cells or the hemocytoblasts, okay, they give rise to other stem cells. So these are like precursors to two different directions, two different stem cell types. Okay, one being the lymphoid stem cells, okay, which will give rise to lymphocytes, or the myeloid stem cells, which give rise to everything else, red blood cells and all the other white blood cells and the platelets. Okay, and so if we look at this in kind of a graphical form or a picture, You can see that hemocytoblast, okay, is going to give rise either to a myeloid stem cell or a lymphoid stem cell. If we go this direction first, that lymphoid stem cell will give rise to a lymphoblast. That lymphoblast still in the bone marrow, okay, will then when it leaves the bone marrow, it's either going to mature in the bone marrow and become a B lymphocyte, or it's going to mature in the thymus. If it matures in the thymus, it's called a T lymphocyte. That's where the T comes in and the B comes in. The B lymphocytes, okay, both of these are part of your learned immune system or your memory immune system. These B lymphocytes will then mature and differentiate into what we call plasma cells later on after they've been stimulated. Okay, and we'll come back and we'll talk about immune function later. If we come back over to the myeloid stem cells, myeloid stem cells could be erythroblasts. If they're erythroblasts, that's the transition. That's the cell that's going to give rise to erythrocytes. And you might remember from previous lectures and other lectures you may have, erythrocytes in mammals do not have a nuclei. So when that erythrocyte leaves the bone marrow, it will lose, the process, the reticuloleucocyte, that process is preparing the cell to become mature. And so the nucleus will be removed, the mitochondria of the cell are removed, and so that's the transition from erythroblast to erythrocyte. Okay, the megakaryoblast, they're going to give rise to megakaryocytes, which give rise to platelets. These are what's going to be the major clotting factor in your blood. The myeloblasts give rise to the granulocytes, basophils, eosinophils, neutrophils, depending on the infection that you might have. You might have different numbers of these. They have different functions. Some attack prokaryotes, some attack eukaryotes, and we'll cover these later. Monoblasts are going to give rise to macrophages, or a monocyte, which then gives rise to a macrophage. Macrophages are like the generalist of the immune system of the white blood cells. Its job is to go around and consume things, break it up, recycle it. Okay, and so they're large cells and they can really consume, pretty much anything. But they also consume a lot of self-made cells. If erythrocytes are old and deteriorating, the macrophages will destroy those and recycle them. Okay. All right, let's look at red blood cells. So they're also called erythrocytes. Often that's the term that you'll probably hear most often from a technical term. They are biconcave, excuse me. Okay, which they have this kind of indentation look to them most often. Inside the red blood cell is going to be a hemoglobin. Okay, and so about 1/3 of the volume is hemoglobin. It's quite a bit, and hemoglobin consists of 4 parts: beta 1, beta 2, alpha 1, alpha 2. Each one of those parts, each one of those amino acid chains, those proteins, okay, that are going to combine together, each one of those can carry an oxygen molecule. So one hemoglobin typically carries 4 oxygen molecules. And you might have enough hemoglobin in a single red blood cell to carry about 1 billion oxygen molecules. Okay, so a huge amount of oxygen can be transported by red blood cells. Now, when there is no oxygen bound to the hemoglobin, we call it deoxyhemoglobin. Okay, if there is oxygen bound, we call it oxyhemoglobin. And this is often when you hear the terms like oxygenated, meaning that the blood has oxygen, is carrying oxygen. So maybe it's coming back from the lungs. We consider it oxygenated. Okay. And then you might hear the term deoxygenated. Okay. That's where these terms are really coming from, meaning it's without oxygen. Okay, back to other components of red blood cells. They lack nuclei. They don't have any mitochondria, so they can't do cellular respiration. They still can do glycolysis, so they still have proteins in the cytoplasm within the cell that allows them to break down glucose and generate small amounts of ATP. Okay, but because they don't have a nuclei, they don't have any way that they can replicate. So when a erythrocyte leaves the bone marrow and is considered mature, it then it's basically its clock on how long it can last starts, okay, because it can't replicate itself. And so in normal circumstances, this is about 120 days, although it depends on the individual. It depends on the activity of the individual, how much use this blood is getting. If you're highly active and you're pushing blood to the capillary beds really a lot, then the lifespan of those red blood cells might be less than 120 days, maybe 100 days, maybe 90 days. It depends on the individual. But at any rate, can't divide, so they can't copy themselves. So when they die or get old, they are decomposed and new ones have to be generated from the bone marrow. Now, on a side note, real quick, okay, often it's thought that the reason for no nuclei and mitochondria in red blood cells is that it allows for more hemoglobin to be stuck in a red blood cell. More hemoglobin means more oxygen transport. Okay? Well, interestingly, we can check to see if that's true because all other vertebrates besides mammals have nuclei in their red blood cells and mitochondria. So we can look at a bird versus a mammal and we can say, all right, well, do birds have less hemoglobin in their red blood cells than mammals? No, they don't. Okay, so is it to really carry more hemoglobin? Maybe that's, I mean, that hypothesis is, I don't want to say it's dead in the water because a bird's red blood cell is a little bit bigger than a mammal's red blood cell on average. So a bird's red blood cell is about 11 micrometers or maybe even 12 depending on the bird. So it's a little bit bigger. Is the increase in size compensating for how much hemoglobin can be stuck in there? Maybe. Okay. One hypothesis is that's why there's no nuclei and mitochondria. The other hypothesis that's out there is typically if there's a nucleus and mitochondria in the cell, it's less flexible and it can't shrink down and fit through capillaries and fit around things like that. So it doesn't have ultimate flexibility. So is that a potential explanation for why no nucleus or mitochondria? Maybe. Birds also have really fine capillaries, and so do some reptiles. So maybe, maybe not. In my opinion, neither hypothesis is really supported by much evidence. And so I think it's still out for debate on why mammals don't have nucleated red blood cells and mitochondria, but all other vertebrates do. Okay. All right, let's dive into the red blood cell count. So real quick, often red blood cells counts are very useful to kind of predict, well, is this individual anemic? Is this individual have a disease? Does this individual have iron deficiency? Is this individual vitamin B12 deficient? Okay, so there's ways at which red blood cell counts can really play an important role in allied health and nursing and other fields that are associated with that. Excuse me. So normally when we talk about red blood cell counts, we talking about how many red blood cells you have per microliter. So It's per microliter of blood, per drop of blood, or however many microliters are in a single drop, depending on the size of the material that you're dropping it with. Okay, sometimes it's considered cubic millimeters. It just depends on what kind of how you're determining this. Okay, so if you're determining it with volume, or if you're determining with an actual like plate reader, it might be cubic millimeters versus microliters. Okay. Any rate, you can make the conversion. You can change them. For males, we're normally looking at about 4 million to 6 million. Well, 4.7. Some people round this up and just say 5 million to 6 million red blood cells per microliter in males. Females more like 4 to 5. Okay, and children like 4.5 to 5, something like that. Okay, it just depends. Again, this is dependent on the individual a great deal, but even more so, it's dependent on where the individual lives. Okay, so if you want to increase these numbers and be at the top end of these, so maybe you're down here because you live in Western Washington and you want your numbers way up here, okay, just move to a higher elevation. You move to a higher elevation, your body's telling the system that there's low oxygen, and it will start stimulating the production of erythrocytes, and you'll start building red blood cells. And then you'll have a lot more red blood cells in solution, in circulation. And this is how a lot of Olympic athletes, marathoners, etc., how they train is they go to really high elevations for, a few months, maybe half a year, stimulate their body to build as many red blood cells as possible. And then when they compete, they're competing at a lower elevation. So they get an advantage because they can carry more oxygen in the system. Therefore, more oxygen, more cellular respiration, a higher metabolism, more energy, etc. So pretty useful kind of manipulation of the body that happens quite often. Again, red blood cell counts diagnose certain diseases. They can also reflect your ability to carry certain, like how much oxygen can you carry. And we'll come back and look at components of this. All right, so the actual production of red blood cells is called erythropoiesis. Okay, so that's the formation of red blood cells. It occurs in the red bone marrow. Okay, and it is triggered by a hormone called erythropoietin. Okay, and that's released, or normally it's abbreviated EPO. It's released by the liver, by the kidney mainly, secondarily from the liver. Okay, and so this will be the result of low blood oxygen level will trigger the release of erythropoietin. That's going to stimulate the production of red blood cells in the bone marrow. The negative feedback to this is high blood oxygen counts, okay, will shut off the erythropoietin being released, and that's kind of the negative feedback. So that would not stimulate the production of red blood cells. Okay, so it takes a couple days. So if you're thinking about doing the elevation training kind of thing, it's going to take you a few days to start seeing a difference in number of red blood cells. Okay, so you move from sea level to 5,000 feet, you're going to get low blood oxygen levels. It's going to trigger the secretion of EPO. That's going to trigger the production of RBCs in the blood, okay, in a few days. Okay? That's why many of them will train months at higher elevations. Okay? Few stages on this. You have the hematocytoblast. That's the stem cell. That stem cell is going to differentiate into erythroblast based on a growth hormone that and erythropoietin will differentiate this into erythroblast. And then you have this kind of stationary, a transitional phase, the reticulocyte. That's where the nucleus and the mitochondrium are going to be removed, dislodged, and it's going to ensure that there's enough hemoglobin's formed correctly, and then once the erythrocyte is formed and mature, that's when that clock starts. Okay, so lifespan's about 120 days, give or take, depending on the individual. It can be a lot less, potentially a little more. Okay, put this in perspective to white blood cells. White blood cells, it depends on the white blood cell also. Some white blood cells, like memory B cells, B lymphocytes, they can last years in solution, and so many years. You can be subjected to a virus a long time ago, potentially 10, 15, 20 years ago, and still carry memory cells to that virus. So red blood cells definitely have a less of a lifespan than many of the white blood cells. Excuse me. So for this erythropoiesis to occur, you need three main things. Okay? Vitamin B12, folic acid. This is going to be required for the DNA synthesis. Okay, so the hemocytoblasts, the erythroblasts, reticulocytes, these all have nuclei. They all are going through the production of proteins and things like that. So remember, hemoglobin is a protein. So these cells are making hemoglobin. Okay, and so that's necessary for the growth, division of the cell, etc. Once it becomes a mature erythrocyte, then there's no more DNA synthesis. So then the vitamin B12 and folic acid is really not that important. Okay, the iron is the important piece at that point, and that's because iron is the molecule that sits inside the heme of hemoglobin. And that's where oxygen binds to, is to that iron molecule. And so individuals that are anemic, they may be anemic because of a lack of iron. And we'll come back and talk about this, right now. Okay, so anemia, or the inability to carry oxygen, or lower capacity to carry oxygen, can be in two main ways. You could have a low red blood cell count. Okay, so you could need a blood transfusion or need to have more erythropoietin generating red blood cells in the system, or your hemoglobin molecules might not be developed because there's not enough iron in the system. So instead of having, lots and lots of hemoglobin within your red blood cells, you might have plenty of red blood cells, but not much hemoglobin to carry the oxygen. Okay. So how does this work? Hypoxia or hypoxemia is low blood oxygen. So when that triggers the production of erythropoietin, normally from the kidneys, secondarily from the liver. That gets in the bloodstream, that gets to the bone marrow. Okay, that triggers, as long as there's B12, folic acid, and iron in the system, excuse me, that'll trigger new production of red blood cells. When you get a bunch of red blood cells in the system, that oxygen level goes up, and the negative feedback stops the erythropoietin from being released, and then you're back into business, and you're not generating lots of new red blood cells. A few disorders, diseases that are associated with this. Okay, you can have iron deficiency. Happens a lot in elderly individuals. Okay, pretty easy to spot under a microscope. Many of you have looked at red blood cells under a microscope. If you are looking at someone's red blood cells and numerous red blood cells have these open white spaces within the system, that means they are deficient of hemoglobin. Okay, so the heme in hemoglobin will give it a color. Okay, and so if there's no color, that's because the individual's lacking iron. So you might have normal red blood counts, normal red blood cell counts, but they might not be very efficient at carrying oxygen because there's lack of hemoglobin. Sickle cell disease, this is a single point mutation. We'll cover it here in a second. But it changes the shape of the red blood cell, which changes the ability of the red blood cell to carry oxygen, to make it through capillaries. And so really what it does is it actually causes the cell to be much more rigid. Okay, so not flexible, because the cell becomes sticky. And so the hemoglobin molecules are actually pulling the membranes in, and it's sticking to the hemoglobin molecule because of a different charge in the molecule, because of the mutation. And so it's less flexible, which means it doesn't move through capillaries and can't get to certain regions, and then you can get regions that are devoid of oxygen. Okay, so how do we treat sickle cell? A few different ways. So again, it's a single base mutation, which codes for the beta hemoglobin, and this will result in a switch in the amino acids. So it's going to switch to a valine amino acid, which changes the charge of that side of the molecule, which means that molecule, it becomes sticky and the membrane gets pulled in, and the cell itself is less flexible. Here you can see that mutation. So here is the DNA sequence. Okay, this is amino acids that are associated with it. It's just a single point mutation. Okay, a single change in a nitrogenous base that now lays down a valine, that amino acid, and you get this long change of amino acids. But all that mutation has to do is change the shape of that protein, the beta protein, and now you have a whole different kind of structure, okay, and which can have lots of problems in certain areas. Okay, because of the shape and the inability to move through vessels and the inability to move through capillaries, often regions will be devoid of oxygen, so it'll start to deteriorate and they'll start to die. The metabolism is breaking down, etc. So it can create severe pain. Metabolism rate also go down. It can also create anemia. So often individuals who have sickle cell are typically extremely fatigued. Okay. So what I showed you before, that little picture of, you notice that some of the red blood cells were normal and then some were sickle shaped. Well, this is very, very common because individuals that are normally battling sickle cell disease are heterozygotes. So half of their red blood cells are normal and half of them are sickle shaped. Okay, so they only have this mutation on one of the chromosomes. On the other chromosome, it's non-mutated, it's normal. Okay, so they produce half red blood cells normal, half sickle shaped. If they were homozygote recessive and both chromosomes have this mutation, the individual doesn't last past really 9 or so, just because the amount of oxygen, the anemia that's going on, the pain, etc., the individual normally doesn't do very well. So individuals with sickle cell will have normal red blood cells and mutated red blood cells. Okay? So how do we treat it? Well, some of the treatments early on is to give things like antibiotics to protect the spleen from infection, because you're constantly pushing, and the blood, these sickle cells can clog and not allow material, not allow white blood cells to get to infections and things like that. So a lot of times antibiotics are given. Fetal hemoglobin is sometimes given. And so sometimes you can get hemoglobin molecules from a fetal perspective a little more efficient at binding to oxygen, and then that can slow down the sickling effect. It can slow down that compression of the individual. In some circumstances, bone marrow is transferred, okay, or umbilical cord stem cells are transferred from other individuals to try to cure the disease. It's worked, except for this is a very, very long process, very painful process, and lots of surgeries. The new experiments that are currently going on and have shown great results. I believe the first person that was done by changing their genome, so just switching this mutation back using CRISPR-Cas9, that was completed in 2012, I believe, and that individual is sickle cell free. And so they just go in and they correct the mutation, and it does involve a bone marrow transplant, but it's a bone marrow transplant where they're transplanting corrected DNA sequences back in. And so the individual in that situation is now making red blood cells that don't have that mutation. And so there's promise, but this technology is super expensive. And so the thing about sickle cell is it's a disease that affects mainly third world countries, or what we might better word for it, developing countries. Many countries in Africa, India, are subjected to sickle cell disease. And so normally a treatment, which might be, I don't know, $1 million, it's just not, it's not efficient. It's not going to happen. Even bone marrow transplants aren't going to happen. So a lot of these others, antibiotics, fetal hemoglobin, might be used to treat individuals with this disease. All right, so to end, let's go through kind of the life cycle and what's happening to red blood cells as they age. So they get old, they're pushed through capillaries, they get damaged, the lipid membrane starts to fall apart, the proteins start to deteriorate, and red blood cells just get old. Okay, 120 days, they're old by that time. The macrophages in the spleen and liver will start to phagocytize these red blood cells. So they'll consume them, break them up into pieces. They break them up into two main kind of things, heme and globin. Okay, the heme is decomposed into iron and biliverdin. The iron can be reused or it can be stored as ferritin in the liver. The biliverdin is often converted into bilirubin, and biliverdin and bilirubin are secreted as bile. So nice recycle system back into digestive juices now and then out of the body. Okay. The globin itself is often broken down into amino acid chains, in which sometimes it can be reutilized to build other proteins, even globin itself. Other times it can be released into the plasma and then eventually excreted out of the body as a waste product. Okay, just looking at hemoglobin itself. So again, it's consists of four polypeptide chains. So amino acids, these are 4 proteins, beta 1, beta 2, alpha 1, alpha 2. When it comes to sickle cell, it's these beta chains that are mutated. Okay, the importance of all of this mainly is that iron molecule that occurs in that heme group. Okay, that's where oxygen's binding to it. Okay, that heme group can then be broken down into biliverdin, which contains the iron, or bilirubin, which loses the iron. And so just different ways to process the material and recycle it back into the system. Okay. And finally, the life cycle of a red blood cell. Remember, we're looking at about 120 days. Nutrients from, obviously, the intestines is going to get into the blood system, be taken from the intestines to the bone marrow. In conjunction with that material, you're going to get erythropoietin coming off either the liver and the kidneys, or mainly just the kidneys, in many cases, that is going to get into the bloodstream and stimulate the production of red blood cells in the bone marrow. When that red blood cell leaves the bone marrow, it's mature, it's a erythrocyte, it's got about 120 days to live. It keeps going through the system, it keeps going through the liver. When they become old, deteriorated, beat up, okay, macrophages will consume them, break them down into hemoglobin, break the hemoglobin into heme and globin. Globin becomes amino acids, heme becomes biliverdin, okay, and bilirubin, and then that can be either processed as bile back into the intestines to help with digestion. Sometimes bilirubin is pushed out in the blood as a waste product. So lots of things going on here, but again, it's a cyclic system, pretty useful and obviously needed for life. Okay, next time out, we'll talk about white blood cells.